Yeah, I'm Ben. This is Bill. And I want to say thanks to Will for the introduction and Tony for the really kind words earlier. It's really great to be here. I think that Bill and I can honestly say that we've really appreciated ASGA showing up and doing everything they've been doing with the resources. We both work on public trust resources. It's not a word that's used a lot with fisheries for whatever reason. BHA, you use it a lot for wildlife, but it should be used with fisheries. And we're out here for the resource, so it's awesome to see stakeholders show up and have their voices heard. Really think it was great for Amendment 7 that people were involved in getting out there. I would say that they brought up recreational data. It's not the same as MRIP, but Massachusetts does have a volunteer angler data collection team, and you have a lot of fishermen and guides here. Probably some of you are already involved in that, but if you're not and you want to be, you can find it on our website. It's the SADCT, which we call SADCAT. For nothing else, but it's a really great program. It helps us get a lot of age and weight data that otherwise we wouldn't have. That goes right into assessment work. And I will also say my day job at the division is a diadromous fish biologist. So I'm not technically a striped bass biologist. And right now at ASMFC, the river herring stock assessment cycle is about to tee up. So forage fish, striped bass, pretty important. That is, if you're conservation minded and you want to see more river herring, please submit comments, go to meetings, make your voice heard about river herring because they're important. And I think we're really at a crossroads. We have been at a crossroads and it's not getting any better. So it'd be great to see people show up for those fish.
Yeah, great. And I'd just like to second what Ben said too, is that I thought ASGA did a really good job getting the public comment out. It's a public resource and it's everybody's fish, but I think the managers appreciated getting that clear message from the recreational sector on your preferences, and I know that it really helped in the process. And so I'm a marine biologist for Division Marine Fisheries. I've been there for over 23 years now. And I wear a couple hats, but I'm a research biologist, and we do a lot of tagging studies for multiple species, not only striped bass, but obviously for the purposes of today, the rest is going to be about some of the research that we've done over the last decade or so in striped bass. So this is pretty striped bass savvy crowd, so I'm not going to bore you with some of the more upper level stuff, but there is some essential life history that should be talked about that is relative to the rest of these talks. And obviously, so striped bass is a large, long-lived species, and their coastal distribution ranges from North Carolina up to Maine, with some isolated pockets even further north and south. And they're anadromous, so they live the majority of their life in salt water, but then they travel into fresh water to spawn. And the males reach maturity between 2 to 4 years, and the females reach maturity between 5 to 8 years. And they have fidelity to their natal rivers, so when they spawn, they'll go back to the rivers that they were hatched in. And so there's 3 major spawning stocks on the East Coast, and that's the Hudson River, the Delaware, and Chesapeake Bay, with some smaller contributions from the Roanoke River. And so striped bass is important to the state of Massachusetts. As Tony alluded to, it's really up and down the East Coast. It's probably the most important recreational game fish, but certainly in Massachusetts. And so by weight, it's our #1 fish that is targeted and caught. And it's by commercial landings, it's not our biggest, but by no means is it inconsequential. The recreational harvest is worth over $500 million in Massachusetts, and the commercial is over $3 million in landings annually. And Massachusetts is also important to the striped bass stock. There's a lot of effort in Massachusetts targeting striped bass. We are usually between 1 and 3 on recreational effort and recreational catch. And then commercial catch, that's a quota-based system, and we're always #3 in landings in commercially landed striped bass. And so Massachusetts has been really active in the management of striped bass, but also in the research of striped bass. So now I'm going to talk about some of our earlier studies that I was a part of, that we conducted back 2006 and on, and then I have a couple studies to talk about, and then I'm going to hand it over to Ben. He's going to tell you a little bit more about what we've done more recently. Let's go back. Right. So in 2006 and 2007, we were receiving reports that there were large aggregations of striped bass in federal waters on Stellwagen Bank. So that was in the EEZ, the exclusive economic zone. And back in the 80s, when stock levels were low, the recreational sector or the federal government prohibited the take and targeting striped bass in federal waters. So these fish were, in essence, not accessible to recreational fishery, and there was this perception that these fish were resident EEZ and never came into federal waters, and therefore the fishermen wanted access so they could get after some of these fish. So we created an acoustic telemetry study that examined these transboundary movements to evaluate, see if they came into state waters from federal waters. And so, so I don't know how much you know about acoustic telemetry, but real quickly, how it works is basically you have transmitters and receivers. And so tags, which is the transmitter, they're surgically or externally attached or implanted into a fish. And that fish transmits a unique ID and sensor information. And when it's within a distance, within the appropriate distance of a receiver, that receiver will record that information until I come or a scientist or somebody comes and downloads that information and that data.
So what you get is like a unique timestamp thing that says this fish with its unique ID was in this place because you know where the receiver was and at that time. So you can actually track the fish that way.
Right. So like I was going to say, this is kind of like Easy Pass. So when you drive through a toll booth and it pings you, that's basically the same type of technology. And so what we did in 2008 through 2010, we tagged 159 fish on Stellwagen Bank in federal waters. And the average size was about 88 centimeters. And we were using a tag that lasted about 2 years. Now the tags last about 10 years, but and they had a range of about 1 kilometer. Yep. And then the receivers were placed strategically in state waters. So when they came from federal waters into state waters, they would be detected. And then we had what we call our gates to the north, so off of Cape Ann, and then south off the backside of Cape Cod, and then Cape Cod Bay. And those were really important, so especially when they moved south, so we could detect when they left and when they returned the following year. And so what we saw after several years of data collection is that EEZ was not a complete refugium for striped bass, and that we saw in unrecorded movements into state waters every year routinely. And these fish would stay there actually for extended periods of time, June through November for some of them. And so therefore these fish were in fact available to the recreational and the commercial fishery. Yep. And so, so after later, when the study was done, we brought in another scientist to assist us that you might hear a little bit more about this afternoon, Dr. Jeff Kneebone, and he helped us synthesize a lot of this information, and that's published in a peer-reviewed journal, so if you want to learn more about the study, it's available. And so we did a good job documenting this longitudinal movement of striped bass, but there was still a lot of unknown about this latitudinal movement, so north to south, and that's important given how important Massachusetts is to striped bass and how important striped bass are to Massachusetts. And so we formulated a question, where are bass coming from, how and when do they get there, and how and when do they leave? And this is important, especially the way if they manage. So obviously Massachusetts has a large impact on striped bass, but when striped bass migrate out of these rivers, they move off the coast, and they're basically treated as one, when in actuality, this coast-wide stock probably has different mortality associated with it as they arrive and migrate at different times. And so this has created a lot of management ambiguity, and so this next acoustic telemetry study was our first attempt to try to address some of those questions. And so one of the benefits of acoustic telemetry and the technology that we used is that once these fish leave our acoustic array and they move down the coast, if someone else is using the same equipment, then they will detect our fish and then they can share that information with us. And so we were fortunate during that time working through the ACT network, the Atlantic Cooperative Telemetry Network, there were receivers up and down the East Coast, and most importantly, there were in the spawning grounds, the Hudson, the Delaware, and the Chesapeake. And so what we saw, interesting, was when the fish migrated up into Massachusetts in the springtime, largely the majority of the fish would come up through the Cape Cod Canal with a smaller percentage coming up the backside of the Cape. However, in the fall, when they migrated south, we saw that largely the most of the fish migrated down the backside of the Cape and down of the Cape Cod Canal. And so that was really important because it showed us the availability of the striped bass to the recreational and the commercial fisheries. And so the other interesting information that we were able to get is the stock composition. Using the detection data that was from the spawning areas, we saw that the fish in Massachusetts during our study time was largely from Chesapeake Bay, but it did vary from time to time. And this conflicted a little bit with some of the previous studies. Bergman and Liebran in 1978 did a morphometric study, which is morphometric is basically when they take a picture of a fish and they take measurements looking for distinctions between different stocks. And so they used that technology, and they found that the striped bass were from Chesapeake Bay, and then Fabrazio in 1987, she did morphology as well. But she found that it varied. It was either from year to year, and it could be from the Chesapeake and to the Hudson River, depending on the year. So, and again, that paper was also published and written up by our core group, and that's available if you want to learn more about it. So those were the two earliest studies that we did, and then shortly after we finished this one up, Ben came along, and I'll steal his thunder, so I'll let him tell you a little bit more how.
Now Bill has to deal with me talking over him at things. So, I had been previously working and done some Hudson River striped bass work with acoustic telemetry, so it worked out really well. I was hired about 10 years ago, actually, just a little over 10 years ago now, division, and Bill had just finished his work with Micah, and we started talking and decided to move forward. So, Will, I designed this so that I would be able to do all the clicking, and so Will and I are going to work on our communication, and I'm going to give his finger a workout. So it'll be a little bit redundant to some of Bill's things, but we talked about taking that last study that Bill just talked about and taking to the next level and looking at what those migrations really meant for mortality and what were bass doing in Massachusetts waters. And like he said, we were able to capitalize, compared, Bill tagged your fish in 2006 or 2009, and then now we're fast forwarding to 2015, and this is actually a diagram showing all the different receivers that fish we tagged in this later study in the mid-2010s showed up on. So we had more receivers, and especially interesting are those off Delmarva. You can see those wind energy area receivers. They're out there testing to see what fish were moving through the proposed wind energy areas. And then also there's a massive gate at the mouth of Chesapeake Bay that wasn't there before for Bill's stuff. But again, it's just a great time to be doing this work. Everybody's using the same technology. And I'm going to cover, I'm going to show one thing about coastal migrations, and I'm going to cover a few other papers that we've cooperated on. And then we'll get into this next big DMF study we've done. Go ahead, Will. Oh, sorry, and just making the point, Hudson River, as Bill made, real quick, Delaware River, Chesapeake Bay, all wired up. All right, so Bill showed the figure everybody's always seen of the coastal migrations with the arrows and the seasons. These are all our bass. And so it's a week time cycle, and the bigger the dot, the more fish were detected at a receiver. So you can actually see through the fall, the fish are in mass. Now they're gonna start coming down, going down through Delmarva off the Chesapeake, spend the winter there. You get to February, fish are gonna start coming into their spawning estuaries, making their way up the rivers. You see in sequence Chesapeake, Delaware, Hudson, coming back down out and into Massachusetts waters. So these are all fish. And that's the diagram. And now we have the power to say when they're leaving, how they're getting from place to place. Because these fish are, for the most part, so coastally oriented. All these people put these receivers out, and we can track individual fish right up the coastline. So there can be holes of 50 to 100 miles, but for the most part, you can get a real continuous track. Go ahead, Will. So that allows us to look at a bunch of different time scales and different aerial scales as well. So this is a paper about differential migration in Chesapeake Bay striped bass. with striped bass, there's been so much research over the decades, and you get one idea early and it never changes, it sticks in people's heads. So this is something where, we're using this technology to change the story because we're finding out more about the bass. 100 fish were tagged over the course of a season, both in the spring and then in the fall through pound nets in the Potomac. Go ahead. And what they found out that, everybody always says, all the male fish stay in the Chesapeake Bay, if you listen to anglers, and they don't come out. And it's the big females go out, but the males stay small and they stay in the Chesapeake Bay. And what they found, and I was a co-author on this, was that migration was size, not sex dependent. And for the time fish were 32 or so inches long, 50% of both sexes had moved out of Chesapeake Bay and were making coastal migrations. And you can see they kind of have the error bars. By the time they're 35, 36 inches, almost all fish are going out and making these migrations. There's a few fish that stay big and don't leave Chesapeake Bay. We've seen it in the Hudson too. But it really is size, not sex dependent. Also interesting is that there was a payoff on that, where mortality was higher for these smaller fish over the time of the study that stayed in Chesapeake Bay. Obviously, there's different regulations within Chesapeake Bay. There's a lot of environmental conditions in Chesapeake Bay that might not be favorable now for striped bass. And they really observed a very high loss of fish from the study in the first year for these smaller fish they tagged. So also very interesting. And then final point is that all the fish that left the bay ended up spending their summers in Massachusetts, getting back to what Bill was talking about how Massachusetts is kind of the epicenter of this recreational fishery. And a lot of fish are over here offshore off Mass or in coastal waters, taking advantage of that bait. You can also look at what's happening and how they're getting from place to place. So this is those wind energy area arrays you could see in the coastal migrations. That tells us about what time of year and which direction fish are going. And you can see on the bottom, so you can see the directions, blue arrows pointing north and then red arrows pointing south in the time of year over 2 years. And the environmental conditions, that array was set up so there was like an inner, middle, and outer parts of the array, and they were interested about how far offshore fish were migrating. You can see the big part of this is the overall pattern, and then you have seasonal patterns on the size and the smaller. And you can see they really had a bit of a sweet spot overall, where they were in that middle. That's the red, that's the migratory pathway they were using as they went up and down. But sometimes they were a little bit further in or a little bit further out, depending on season. So these are all the kind of really important things that you can then take to management when you're designing a wind array. Where do you want to put things? Where do you not want to put things that acoustic telemetry and other biotelemetry techniques can get you? And I'll take a quick dip into the Hudson. This is a really cool study, and what was found was that within that spawning population in the Hudson, there's actually different what are called contingents. So there's a group within the same population that's acting, has a different set of behaviors and can lead to a whole different exposures to fisheries and other things. So what they found was that there was a whole group of slightly smaller fish that were spawning lower in the Hudson than the bigger fish that were going all the way up by Albany. And you can see this is how that shows that you have the red, the higher river spawning group, and then that blue lower, and that dark black line is the centroid, is kind of the median of all these spawning behaviors for the two groups. You can see there are fish that do other things, but that's the median behavior. The red on the lower right are fish that were tagged in the first year in that lower group, but then the second year went up to the upper region. So these kind of oddballs, and behavior does change as fish change sizes as well. But what they also found is that could lead to differences in mortality. So these are fish loss in the 2 years in 2017 and 2018. And what you can see in 2017, what happened here is that the lower river warmed up quicker than the upper river, and that lower river group dropped out of the river after spawning about 2.5, 3 weeks earlier. So like early May, they dropped out of the river, and it looks like they were basically some of these first fish that got into the Long Island, mouth of the Hudson fishery, coming out, and they just disappeared from the study. They were gone. And you think about us right now. I'm a striped bass angler. I'm pretty excited to go striped bass fishing when my river herring work ends. But a lot of guys are out there right now, and you get people, effort's high right now. People are excited to go fishing, and this is that first slug of bigger fish that hit the water, where you're getting caught a lot, and probably led to increased mortality on that group of fish. So all of a sudden you have this behavioral attribute of a population. They're different than the rest of the fish, and that's going to lead to them getting more mortality. So these are the kind of things you could never get at before you had tools like biotelemetry. So I'm going to go and talk about this next project that Bill and I did with Mike Adine and Mike Armstrong. And I'm wrapping up the acoustic telemetry analysis over the next year or so and working into the genetic stock ID part, but I'll talk about both things. So this is a very similar technique as Bill quickly went over. This time we went for 3 different study areas because we wanted to see if fish behave differently and had fidelity to not just their spawning area, but to places they spent the summer in Massachusetts waters. So we made 3 study areas: Boston Harbor, the backside of Cape Cod, and then where we are now, really, Buzzards Bay and Vineyard Sound. And we tagged almost 260 striped bass. We tried to evenly tag in all 3 areas in 3 separate size classes. So we had at the time, and this is great, you have a decade of consistent management levels. We were joking about this on the way down and where fish were. So at the time in 2015, it was 28 inches below 28. It was going to be a schoolie, and you're putting it back. And then between, you had the slot of everybody could keep from 28 in Massachusetts from 28 to 34. And then above 34 was a commercial fish. So we had our schoolies, our recreational fish, and our commercially sized fish. And we wanted to know across those size classes if mortality was different. We used the same tag Bill was using before. At this point, it had a 7-year battery life, so we could follow fish for a long time. And so this is what our array looked like for this study. Anything red, Bill and I were maintaining out of Gloucester, and then the blues are Greg Scomel's white shark. But you can see that we gated off Boston Harbor. We had a gate off of Gloucester, a gate off of Peaked Hill off Provincetown, and then we completely gated off Buzzards Bay and Vineyard Sound. They're all those red receivers, and the Cape Cod Canal as well. And our idea, why we really started thinking about this, was harvest hotspots. For well over a decade, what we had seen was the escalation of the recreational commercial fishery on Cape Cod, here on Cape Cod, and the Nauset, Chatham Bight, and everything that was going on. And you can see commercial landings here over 25 years. Go ahead, Will. And what you saw was the pattern of everything in the north in Buzzards Bay going down, and then the Cape Cod landings just going up and up and up. It's a little bit of a different story now. We didn't really need this figure to show us what was going on, Will, because this is what we were seeing on the water and the reports we were getting. A lot of the folks down here probably had to live through this, but it was guerrilla fishing, and still is at times. So we were concerned about, if there's this really intense fishing, we're moving a lot of biomass in one place at one time, could this be having an adverse impact on certain stocks over others? And that was a big part of what we wanted to know. And as Bill said, is we're doing our research to resolve management ambiguity. We have all these rivers, but we manage them like this, so that leads to striper pie. We don't know what's in it, but it's a pie full of stripers, and that's what our harvest is. So we wanted to provide information on stock composition of aggregations, examine whether those were stable year to year, were the same fish going back to the same place every year, or were they going different places? And then to determine where a fish came from, how it got there, and where it spent its summer in Massachusetts affected the chance it was going to end up dying in a fishery. Like, how does that affect the fishery mortality? All right, I'm going to get up really quickly and explain this, because I'm guessing nobody here, maybe 1 or 2 people, have seen what this is. This is a chord diagram. And so on the top half here, you have aggregation. That's where we caught this fish. And you can see there's Buzzards Bay, Cape Cod, and Boston Harbor. Those are the 3 places we were tagging fish. On the bottom half, we have the spawning area, and this will change from figure to figure, but it's the same idea. So each little sliver, like these little slivers, that's one fish. So on the top's where we caught them, connecting to where they ended up going is the spawning area. Everybody get it? Any questions? Okay, good. Yeah, no, they're confusing when you don't. I think they're very cool diagrams, and they can be very powerful, but they're confusing the first time you see them. So the thicker that whole band together is, the more fish, right? So what you can see here is we tagged 175 bass, and we were tagging across all size ranges down to like 20 inches. So we only got 70 fish that ended up being detected between 2015 and 2016 in the spawning estuary, but the majority of these fish were located in the Hudson River during the spawning season. And this is true for all 3 of the aggregations that we were tagging in. And there was really no difference. And there's the fancy scientific test to tell you so. Okay. So So looking at the fish we tagged in 2017, fairly similar story. We did manage to tag more fish from the Delaware. We did have 41 fish that were tagged in 2015 and 2016 that showed up in the estuaries, and I'm having so much trouble. And there were 3 fish, which was cool. That's something we don't know that much about, that went from basically 2015 tagged fish, then 2016 were in one spawning estuary, and 2017 were in a different spawning estuary. So the ghost of Frank Zappa is here to remind you that the action's really out there on the edges. The medians are fine, but it's really cool what's happening out there on the edges. So it's interesting to see those patterns, because I mean that's how you have, and in the big picture I'm making a joke, but that's how you have new rivers start. You know, like these rivers where we don't have spawning now, it's those spillover fish that are going to go and be exploratory and go spawn somewhere where habitats opened up. They're going to reboot populations. So that kind of behavior is important. Again, majority of the fish located in the Hudson, and fancy scientific tests to say there's different aggregation composition, sorry. All right, now 2016 and 2017, we have different connectivity. Again, that Delaware is really what did it. Go ahead. And now let's look at a size effect. Did size make a difference? So we have on the top our smaller fish, our schoolies, then our rec fish and our commercial fish. And in 2016, there was a difference. You can see as the bigger fish, there was a stronger contribution of Chesapeake fish. Those big fish we were tagging, more of them were Chesapeake. And we saw the same thing in 2017. I'm not sure whether this is about size differential migrations, if like different populations are doing different things in their migrations, as much as it is that we were tagging in 2015 and 2016. And at that point, it had been a solid 10 years since there had been a good Chesapeake year class. And the smaller fish were just more proportionally Hudson by this point. And we were still seeing in those larger fish that we were tagging that were 8, 9, 10, 12 years old, a fair amount of Chesapeake fish, because that went back to the last good year classes that were coming out of the Chesapeake. But something I want to look at a little bit closer. So year to year, do fish do the same thing? How many of y'all are anglers? How many of y'all think they do the same thing? 1, 2. You're also a scientist. You don't count, Luke. You count. Sorry, I'm just kidding. Okay, so here we go. Bass tagged in 2015 during 2016. So you can see, go ahead, Will. These are the 3 tagging areas we had. Because of the receiver coverage, we could blow it out. So that was saying if we tagged a fish in that small area and it showed up in this larger area, that's what we're going to say they were seeing in the same area from year to year. And this is what we saw. So on the top, these are fish we tagged in 2015 that came back in 2016. So we handicapped ourselves, and we didn't start tagging until about July 15th, and we stopped tagging usually by like September 5th, September 10th, because we wanted to get fish where they were hopefully set up for the summer, which made great fishing in August, always the best, bass fishing in August. But what you see here is that for the most part, fish really went back to where the same general areas in the second year where we actually tagged them. The lowest number there, that's 70% for Boston Harbor. I actually will probably end up lumping the North Shore, like that north of Cape Ann, into Boston, like just saying the whole thing is north of the Cape Cod Canal, because you see a lot of behavior where they kind of skip back and forth. When you look at the actual daily records, you're like, all right, it's up here for 2 weeks, and they came back down, then went back up. Go ahead. 2016, same thing, tagged in 2016 during 2017. Yep. Again, very similar results. Now this is where it gets even cooler. All right, so if this is a fish that we tagged in 2015, where did it go in 2016, and then where did it go in 2017? And if it was a 2016 tagged fish, where did it go in 2016 and then in 2017? So we let the fish tell you where it wanted to be. Say that first year at large is where it actually wanted to be. We just caught it where it was going to. And what you see there, you already did it, yep, is that it's even higher when you do year 2 to year 3 of that fish being at large. One other way to look at it is fish with 4 years at large. And so basically this is a bar chart. The bigger the bar, the more things there are. And if it's a color, it stayed in the same place the entire time for the different regions. And what you see is that 90% of those fish we had this long of a of a time record for stayed in that same area over that entire time. They didn't switch area. How are fish getting from one place to another? That could also be important. So I am going to get up again really quickly, just because the text is small. So the first thing is where they came in the spring. Second, where they went in the fall. So last one is fish that just came into Buzzards Bay, left Buzzards Bay. They never went anywhere else. The next one is going to be fish that came in the spring, as Bill showed before, through the canal, went back out through the canal, and hopefully everybody can read everything there. And what you can see, and then the color coding is by where they were tagged. So what you can see is those Cape Cod fish, and Bill, importantly, his tagging in that first study was off of Cape Cod. It was in federal waters, but it was up on Stellwagen off the just north of Cape Cod. And here we tagged fish from all over the different places, and we're seeing something different, where these fish that summer north of Cape Cod, or of the canal, almost always go through the canal in the spring and go back through the canal in the fall, unlike those Cape Cod fish that go down the backside in the fall. Go ahead. Saw a very similar pattern in 2018. There's some extra rows here, and this is really cool, and this is why I love talking to people down here who fish hard, because especially when I saw that second one, the third one, where it's all Cape Cod fish, the pink, and it says Buzzards Bay, Vineyard Sound to the Cape. Like, those were fish that I've only seen it in that year, where they came in through Buzzards Bay, and then they went out through like Quick's Hole or Robinson's Hole or Woods Hole and out around the Cape. And I could not figure out what was going on, but Will Poston mentioned Brian Coombs. He told me he'd seen the same thing. I ended up talking to a bunch of anglers down here, and they said that in the spring of 2018, there were a ton of squid and butterfish in Vineyard Sound. So my guess would be, again, is fish are going to go where the food is, and they went and found all that bait and went out and around, which is a really interesting thing to then see that happen with the behaviors. But again, in the fall, those northern fish are going up through the canal, down through the canal. So how does that link to mortality? Putting together these whole big picture, that's the whole end game for us. As an example, in 2017, almost 30% of our tagged bass that entered Massachusetts waters in the spring, that's where the last time over the course of the summer they didn't leave. 30% of what came in didn't leave. And so when you look at that, now we have a bar chart, and on the bottom you can see where the fish, the last detection for the fish was, and then the color coding is by season. So green in the spring, red in the summer, and orange in the fall. And not unsurprisingly, the canal, there's a lot of fish. The last time we heard them was in the canal. If you're from Massachusetts, you know that the effort's a little high in the Cape Cod Canal. I saw only one guy when we came over the Bourne Bridge today, but it can be pretty high. And importantly, how that all translates out is, that all of that fall canal effort, which is like our biggest single thing of fish dying, was from fish that spent their whole summer north of the canal. So there is potentially some real differential mortality on fish depending where they go and how they get there. So quickly, I'm going to talk about the genomics portion of the study, and that data is being processed right now. I don't even have the data in hand. All the samples are up in Canada being run over the next month or two. So I'm really excited to get at the mixed stock samples, I should say, not necessarily the other work we've done. So I'll go over to some of that. Anyways, anybody my age here right now is hopefully smiling at this slide. This is a good question for genomics. It's not genetics anymore, it's genomics. We're looking at these huge data sets. We have a lot of power to do things. So we're hoping to use these tools, and I'm going to go over really quickly, both for this project and then to inform future stock assessments, and knowing is half the battle. So what's going on with genomics is they're just getting so fast and so powerful. I mean, if the COVID vaccine thing is really the same thing that allowed us to have COVID vaccines as quickly as we did, is the same thing that I'm using here now. It's the same technologies. And so all of a sudden, in the last decade, we've gotten the capability where you could literally collect samples from a fishery on a Tuesday, and by Friday, you could know where those fish came from if you have all the information you need. So all of a sudden, you have this technology that's going to give you reproducible from lab to lab results, fast, so you can actually make informed decisions about the information you're collecting, and they're going to be accurate enough for management use. So genomics has really turned a corner in the last 10 years. And this is an example of an Atlantic salmon paper. And so you can see all these the different colors for different runs. And in the lower right-hand corner, there's like an arrow pointing towards these little islands. And that's where this fishery actually occurred. So go ahead, Will. And what they found was that most of the fish they were catching weren't from anything we were near there. They were, in fact, a lot of them were from northern Labrador, but they were able to figure that out through genomics and figure it out quickly so they could actually effectively manage this Atlantic salmon fishery that was an ocean intercept fishery, and they were catching salmon from everywhere. They're doing this with a bunch of salmonids on the West Coast. We're starting to hopefully get it not on a real-time basis, but pulling the oil stick on river herring and other bycatch species. So the idea is now to bring that to striped bass. The first step in that is figuring out what the genomic blueprint of striped bass looks like on the East Coast. So 2 years ago, we published this paper, and this is where all the populations on the East Coast we sampled from. So we had a really good spatial and fairly comprehensive as to where fish are spawning. Baseline is what this is called. And this is what we found. This is another hard to interpret figure, I apologize, but basically what you're seeing is if it's the majority of the color in that section it means it's different. And what you can see here is that the first three groups there, 1, 2, and 3, are very distinct. Those are all Canadian, and you can see how solid they are for the Miramichi, Shubenacadie, and St. John River. And then the fourth group is the Hudson, and it's a little bit more muddled with other things. And now you have the Delaware and all the Chesapeake runs, which are a lot more muddled. But they are a distinct group, and the Delaware does fall in with those Chesapeake. There is some ability to differentiate between Western and Eastern Chesapeake, and then we have the North Carolina populations. So when we go to assign a fish in the future, these are the groups we'll be able to get back to. It'll be like stock complexes, they call them reporting groups, but we aren't able with the current technology, and I don't know if we will be able to get right back down to an individual population. Part of that is because we've done so much stocking across places. So when we take a fish from the Chesapeake or from one run in the Chesapeake and then reseed a different river, all of a sudden that they're going to look the same genetically. This is the same way that we did the Hudson up to the Kennebec. The Hudson and the Kennebec are different populations. They're going to be working differently. But because we seeded the Kennebec with Hudson fish, using these tools, they look the same. And then going back to the accuracy, what's really good, this is basically a cross test, is that if you basically played with the sample sizes and did a bunch of millions of tests, what you see is that it's very good at getting fish that were, these are known population fish. These are from our baseline. So we had, 247 fish in the Delaware Chesapeake group. 247 of them ended up being assigned back to the Delaware Chesapeake group, and one fish where they just couldn't assign it. So this is a really high accuracy. It's over 90% accuracy overall, 95% accuracy. So it's going to be a great tool. We have over 5,000 samples we've collected through Bill's group, his fisheries dependent investigations, and then through SADCAT angler program I was talking about and we're getting those run right now. And I do want to, for all the other Massachusetts anglers, thank you, because that's recreational license money that helps seed that. So we run a bunch of them on recreational license money, and then we use that as match to get a federal grant. And the deliverable on that federal grant should be that we're going to use all of the 5,000 samples to come up with an empirical model on how many samples and how often every state would have to take to get a good estimation of the reporting group level harvest in coastal waters, so we can finally get away from that striper pie and have actual mortality attributed to each population for the coastal harvest. So hopefully in the next 5 years, that's what happens. And hopefully next year we can come back and I'll be able to tell you more about that and what that actually looks like. As I alluded to earlier, you make these plans, you do these big studies, and then everything changes. This is just when you have some stability, it changes. So I'm going to hand this back to Bill, but I think as everybody knows with Amendment 7 and a lot of things Willie and Tony and everybody else and John have been doing the good work on, it's been a changing fishery. So this is just a figure to show that in the last 20 years, we've picked up a ton of recreational landings and as the stock has gone up, and recreational release mortality has become a big issue. And in Massachusetts, go ahead, Will. That's definitely a big issue because, as Bill showed earlier, we're typically second or first in the amount of recreational catch in the state. So that ends up being a lot of recreational discards and a lot of dead fish that we didn't mean to be dead, but unfortunately, for whatever reason, they end up as dead fish. So, yeah, it's the next problem.
All right, so not to keep you from your lunch, but I did want to just touch on real quick on some of the studies that we're currently working on and I think you'd be interested in. So this is a study that we've done over the last couple of years, and it's use of the circle hooks as a conservation tool in the striped bass fishery. And so now that, the common theme here is we know the condition of the stock right now, and the SSB for striped bass is decreasing, and Massachusetts harvests a lot of striped bass. In 2019, we sold 193,000 saltwater fishing licenses, and we estimated that we caught over 5.5 million bass. 195,000 of those were harvested. And so if you apply that to the currently used post-release mortality rate of 9%, that's like 500,000 striped bass dead. And so when we saw this trend happening, Massachusetts got proactive, and we mandated the circle hook. So anytime you're targeting striped bass, you need to use a circle hook. And then ASMFC the following year mandated it coast-wide. And why use circle hooks? It makes a lot of sense. They've been widely studied in other fisheries. The Billfish Foundation requires them. They're federally mandated if you're offshore shark fishing. In Florida, they use them for reef fish, for targeting groupers and snappers. But surprisingly, there was a paucity of information on the use of circle hooks for striped bass. So using acoustic telemetry, we created a new study to estimate their conservation benefit. And this made a lot of sense for us. So based on Micah Dean's and my past experience, we've studied post-release mortality in other fisheries, and we've done it for ground fish, mainly haddock and cod. And it was easy then. We put out an array of receivers. We monitored their vertical movements between receivers and then their horizontal movements by depth. And so in the bottom right, if an active fish was swimming around, you could look at his depth profiles and his movement. That's on the live fish. The one on the bottom, you can see where it basically flat lines in depth, only moved one receiver, two receivers. That was a dead fish. But technology's evolved, and so now what we applied was a little bit different tag, same company, same concept, and that was the use of an accelerometer. And these can be used to measure acceleration or tail beat, and we chose to use tail beat. So the way this works is that basically instead of measuring movement on all three axes, it just measures on two axes. So as the fish is swimming, it's recording that pattern. And so there's some data on the bottom right, and that's what it looks like when you plot it. You have a meters per second, so that's your speed over time. And then if the fish dies, you'll see again, it's very similar, it flatlines. So this is a really easy way for us now to tease out mortality. What also is really cool about this tag is that you can get mortality over a longer period of time. So rather than like, if you think you catch a fish and it doesn't look good and you throw it back, you assume it dies, a couple hours? Well, a lot of our fish died days after they were released, and you can see they slowed down and then they slowly succumb to that release event. And so this was a really unique tag that we took advantage of to do that. So over the 2 years, we did 4 treatments. We tested the 6.0 octopus J-hook, and then a 6.0 inline circle, an 8.0 inline circle, and then the Eagle Claw 10. And we're just trying to detect the difference between the hooks and then between the hooks and the J. And we used only natural baits. We didn't do any artificials. We used just live or dead bait. Most of the time it was mackerel because that was what was available to us. And then we also collected other data elements that will be useful probably down the road, such as temperature, angler skill level, fight time, handling time, and release time. And so the other really interesting part about this study for us is that when we caught the fish and released it, we assigned a score to it. So, for example, if it was a perfectly hooked fish and it was caught in the hinge, say, we'd call that a condition score 1. But conversely, if there was a fish that you got it and it was almost dead on arrival, or looked like that on the bottom center picture there, mortality was imminent, and you knew that was going to be dead, and so we could assign that a condition, a score of like 4. And so what's interesting, what will be useful for that is that right now we use acoustic telemetry to associate mortality to those condition scores, but in the future we could come up with a study design, maybe ask anglers like you to document how the fish was caught, and then based on just the location and the condition of that fish, we could assign mortality to that fish and not even have to put a tag in it. So the circle hooks improve survival. Unfortunately right now it's inconclusive. We are working up the data this summer, and we hope to have something out in this fall. But as Ben and I can both attest, and I'm sure many of you anglers out there that use circle hooks now for striped bass, they're really not perfect. And there's probably other factors that are going to be contributing to post-release mortality, such as the size of the hook, the handling practices, and the temperature and water. And so moving forward, circle hooks may be a really good tool, but education really is going to be paramount. And this is kind of probably going to lend into what you're going to hear more about this afternoon after lunch from Keep 'em Wet. But handling practices, better handling techniques, and really how to reduce these fish. Because, effort controls and these hooks aren't going to save this problem now. We really need to find other ways to reduce mortality in this fishery until we can get some good year classes coming back and rebuild this stock. So with that, that's it, I think, for now. And if you guys have questions, we'd be happy to answer them.
For those who need water to breathe, Costa sunglasses.